So as I mentioned in class, we won't really be focusing very much on meiotic recombination, and we're going to instead talk more in depth about the mitotic recombination and why that might happen. Mitotic recombination and replication is going to, or during replication, is going to take care of bulky adducts or single-strand nicks. When these are encountered by the DNA replication machinery, it can't continue on through, and so it stops. I think I used the phrase freaks out in lecture. And so we need to actually have a mechanism to fix these if they haven't been previously fixed. And so this is to keep replication going. So the first one that we've got is replication fork restart by homologous recombination. And this is the result of a collapsed fork. This is when there's a nick on the template strand. So if we have a nick on the template strand, in this case it's the lagging strand, we're going to wind up with what looks exactly like a double strand break here. In this case though, we're not dealing with a homologous strand, we're dealing with another strand that's already been synthesized. So to fix this, we have our double strand break here. We start out with strand invasion, and just to orient you, the blue strand is still the blue strand. These two just have been flipped from here to here. So we have strand invasion, and what this is doing is it's using the complementary strand. So just as a reminder, this is complementary to this here, just as this is complementary to this, and this is complementary to this, and this is complementary to this. It's important for us to recognize those because we're not using a homologous strand, we're using the other template strand to fix it. So here our 3' end will invade down here, and we'll add on, we'll use this now bottom strand here, which formerly it was in double-stranded DNA with. It will use that as a template to make a bit more of, to synthesize a bit more of DNA on the 3' end. Alright, so this is originally where our strand break was. So we have an invasion, creation of the D loop from the new strand, and then extension of this one. This causes a holiday junction, which will need to be fixed or resolved, and so we have resolution of holiday junction. There's a little bit of the new strand, the old strand that's now associated with this new strand here. We have the crossing over or homologous recombination event here, which winds up getting us back to this point where we now are connected to our original template strand here. And so this means now that we can continue on with our replication from this 3' end, and in our lagging strand we just lay down a new primer here and then continue with the synthesis. So that's a replication fork restart based on a nick in the template strand that winds up causing a double strand break, and then the actual fixing of it is almost exactly the same. Restart by homologous recombination when there's a stalled fork. So this is when there's a big bulky adduct potentially right here. This is our adduct. And so it stops the, in this case, the leading strand from being able to make more DNA in this direction. So the replication is stopped here. Now, what's important to remember is that this is in the same place on the DNA strand. It's not moving around on the DNA strand. This is the same base because we start out with a branch migration. And so this, which used to be in the open part of the fork, is now re-annealed back here. So this bit is this part that we're seeing here. And what happens is that as this strand and this strand re-anneal for this branch migration, this strand and this strand, the newly synthesized strands, have sequences that are complementary to one another because they're based off of sequences that are complementary to one another in this template strand and this template strand. So as this part here re-anneals with this part here, there's a bit of sequence here that can re-anneal with this sequence here, and that's how we wind up with this part in here. So we've got a bit of a 3' overhang, or sorry, 5' overhang, because there was no stalling on the lagging strand. So this is where our leading strand stalled. So these base pairs here are complementary to these base pairs here. So I hope that orients you a little bit better to what we're talking about here. So once this branch migration has occurred, there's now a little bit of a 5' overhang that this leading strand can use as a template to make more DNA. And what is actually happening here is we're making more DNA that's complementary to this lagging strand, that's also complementary to this sequence here that goes over that bulky adduct. Okay, so this is going to basically leapfrog the whole replication machinery over this bit here. So what happens then is, instead of having the branch migration where these parts anneal, now we have the reverse of that branch migration. So it's going to migrate this way, and we wind up getting the reopening of our replication fork to where it had been opened before. The only difference now is that whereas we had stalled our replication machinery right here at the beginning of this diagram up here, now we don't have to even deal with this bulky adduct because the replication machinery is going to start beyond it, and then we can just have the replication fork continue on. Finally, we have something about double-strand break repair without homology. So in our previous versions, we've talked about that it has recombination, no recombination, but it's always using a homologous sequence in order to actually repair the double-strand break. And in this case, we're going to be using no homologous sequence to help. So we think of this as being in a case where there's no homology or microhomology of less than 10 nucleotides. It's also called illegitimate recombination, which I think is really funny. But that's because there's no homology used for this recombination. So non-homologous end joining begins with, of course, our double-stranded break. And so this is just showing you that there's a double-strand break. And there's proteins that are going to recognize these ends, just as in our normal double-strand break repair, only these are different proteins. So it's the Ku proteins. The Ku proteins will bind to the ends, and it'll bring it close together. Once the ends are brought close together, MRN or MRX, which are the proteins that are involved in double-strand break repair processing in humans for MRN or yeast for MRX, are going to actually form a bridge between the ends here, leaving us with two single-strand nicks, one on either side. Then, of course, we're going to need a ligase, and the ligase comes in, seals those nicks, and then we wind up with our repaired DNA strands. So again, it's not necessarily a very complex process in terms of what we're showing here, but the important thing is to recognize that it is possible for end joining or double-strand break repair to occur without homologous sequences to provide that basic bridge there. It can happen that proteins will facilitate this process.